everyone, welcome back to BIOSKUL. This is Dr. Gayatri. In the previous video, we have discussed question number 1 to 15 from Part B evening session of CSIR NET examination 2020. In today's session, we will discuss question from 16 to 30. So let's get started. Which of the following is not released by sympathetic preganglionic neurons? So it is asking about which is not released by the sympathetic preganglionic neuron. Neurotensin, it is released by the sympathetic preganglionic neuron. Enkephalin also. But the serotonin, it is not released by the preganglionic neuron. Substance P, it is also released by the sympathetic preganglionic neurons. So the correct answer for question number 16 is option 3, that is serotonin, which is not released by sympathetic preganglionic neurons. Next question. In agrobacterium mediated transformation, which one of the following approaches is more likely to generate transgenic plant with incomplete transfer of passenger gene? Okay, it is asking about the incomplete transfer of the passenger gene. So in the agrobacterium transformation, there the tDNA which is transformed or that is integrated into the infected plant DNA. So that portion which is remain in the left border that may not be transferred completely. So the passenger gene, if it is present in the left border, then that may lead to the incomplete transfer of the passenger gene. So let's see the option. Option 1, placement of selection marker gene toward the left border and passenger gene toward the right border of tDNA. If passenger gene is present towards the right border of tDNA, then it will be completely transferred. Okay, so this one is wrong. Next question. Expression of selection marker gene under constitutive promoter and passenger gene under tissue specific promoter. So expression of selection marker gene and the transfer of passenger gene is not related, so this one is also not correct. Okay, let's see the option 3. Placement of passenger gene toward left border and marker gene towards the right border of tDNA. Okay, if we place the marker gene toward the left border, then it is more likely to generate the transgenic plant with incomplete transfer of passenger gene. So option 3 is the correct answer. Let's see the option 4. Expression of both selection marker gene and passenger gene under constitutive promoters. No. So the correct answer is option 3. Okay. Next is question number 18. A lectotype refers to. Okay. Lectotype refers to the option 1. It is a specimen of the opposite sex to the holotype and designated from among paratypes. It is not lectotype. It is known as the allotype. Okay. A specimen of opposite sex to holotype and designated from among paratypes is known as the allotypes. Option 2. An illustration based on which a new species is described. A new species. New species means it is holotype. So this one is also not correct. Okay. Option 3. A specimen later selected from a group of syntypes. Selected from a group of syntypes to serve as the specimen for a species after its original description was published. So this defines the lectotype. Lectotype, it is the specimen which is later selected from a group of syntypes to serve as the type of specimen for a species after its original description was published, which is known as the lectotype. So the correct answer is option 3. So let's move to the option 4. A substitute specimen selected to serve as the type of specimen of a species after its original description was published. Okay, it looks like the lectotype, but see, when an original holotype has been lost or destroyed, here the original holotype is lost or destroyed. So if the original holotype is lost or destroyed, then it refers to neotype. It's very important to practice the previous year CSIR net question, because though you did not get any direct question, but you will get some related questions. Okay. Next moves to question number 19. The immunoglobulin heavy chain that is rearranged fast and is displaced on the surface of early stage of B cell development. So it is considered the B cell development, and in the B cell development what happened? As the heavy chain is first rearrangement started, then followed by the rearrangement of the light chain. So it asking about this heavy chain rearranged fast and is displaced on the surface of early stage of B cell development is associated with. So heavy chain is obviously associated with the light chain. So let's see the options. The option 1, class 2 associated invariant chain peptide clip. Clip, it is associated with the MSC, major histocompatibility complex. So this one is wrong. Then a surrogate light chain, yes, this one is correct. Heavy chain associated with the surrogated light chain. After the rearrangement of the light chain, the surrogate light chain will be replaced by the light chain. Then the option 3, it is the beta 2 macroglobulin. Beta 2 macroglobulin, this is associated with the MSC. So this option is also wrong. Let's see option 4. Immunoglobulin like cell adhesion molecule. It is a cell adhesion molecule. It has no role in the B cell. So this one is wrong. So the correct answer is option 2, that is a surrogate light chain. Question number 20. Which of the statements given below is incorrect? Okay. So it is asking about which one is incorrect. Let's see the option. The three common types of membrane lipids are cholesterol, phospholipids and glycolipids. We all know that the three common lipids which are present in the membrane are cholesterol, phospholipids and glycolipids. So this one is correct. Okay. Option 2. Phosphoglycerides carry glycerol backbone to fatty acid chain and phosphorylate and phosphorylated alcohol. This phosphorylated alcohol, it may be the choline or it may be the ethanolamine. So this one is also correct. Okay. Option 3. Most phospholipid and glycolipid, they form bimolecular sheets. They form the bimolecular sheet rather than micellin in aqueous medium. Okay. This one is also right, because the phospholipids and the glycolipid in the aqueous medium, they will form the structure which is like this, like a bimolecular sheet. This is the bimolecular sheet, but in the micellin there is the presence of the single tail like the structure. So this one is also correct. So most phospholipid and the glycolipid, they form the bimolecular sheet in the aqueous medium. Okay, option 4. The common alcohol moieties in phosphoglycerides are glycerol. Okay, in the phosphoglyceride, they carry the glycerol backbone, fatty acid chain and a phosphorylated alcohol. So the phosphoglycerides, the alcohol moieties, it may be the glycerol, it may be the inositol, sometimes it may be the choline or it may be the ethanolamine. But it never be the tyrosine. So this one is wrong. So the correct answer for this question is option 4. Okay. Now move to the question 21. Human polysyndactyly. Human polysyndactyly, that is the joining of the extra-digit syndromes that result from a homozygous mutation 8. So it will be because of the mutation of the Hox gene. Okay. Hox gene. So there is no involvement of the antennapedia complex. Okay, antennapedia, it is related to the Drosophila. there is not the beta catenin locus, so it may be the HOX-D or HOX-C. So for this question, human polydactylite syndrome, it result from the mutation of the gene HOX-D. Okay, I have a video on this. You will get the link in my description box, or you can go through my YouTube channel BIOSKUL. Question number 22. Which one of the following statement forms a chemical mediator that is involved in the mechanism of pain during inflammation? So when it comes to the pain, the one thing that comes into our mind is kinin system. Okay. Kinin system, it is related to the inflammation. So what is the correct answer? That is kininogen and the bradykinin system. Okay. If you don't know the answer, then if you analyze all the options, then also you can find out the correct answer. Okay. The option 1, activated blood clotting cascade. This is not related to the inflammation. Plasmin fibrinolactic system. This is also not. B cell activation and the inflammation. These are also not related. The correct answer is option 3. Question number 23. In the enzyme-linked antibody used in ELISA, the interaction between enzyme and antibody is stabilized by. Okay. So, in ELISA, the enzyme and the antibody, they are always linked by the strong covalent bond, right? They are linked by the strong and the covalent bond. Here you see hydrogen bond, ionic bond and van der Waals interaction are the wrong option. So, enzyme, it is bind to the antibody by strong interaction, that is covalent bond, so that the antibody should not separate from the enzyme. The correct answer is option 3. Okay, let's move to question number 24. If the pyrrolidine ring of proline is reduced to a linear form, the new amino acids will have. Okay. So, what is the structure of the proline? Proline is nitrogen and so this is the pi bond and this is psi bond and the pyrrolidine ring. Pyrrolidine ring or the R chain, it is associated with the amino group. Okay. So it says that if the pyrrolidine ring of proline is reduced to form a linear form. Okay. If it is reduced and it forms the linear structure. Linear structure means it forms the linear structure. The new amino acids will have constant pi then proline. Okay. So if it form the pyrrolidine ring, then it attached to the amino group or the alpha chain, so it will affect the pi chain. There is no effect of psi chain. So the constant with the psi then proline, it is wrong, and this one is also wrong. So constant pi then proline, no. If it will be the linear in form, then that will be relaxed. So the correct answer is relaxed pi then proline. So the correct answer is option 3. Okay. Next move to the question number 25. The following table lists name of the scientists and advances made by them. Okay. So the here option A, Linus Pauling. So when the word the Linus Pauling is comes, then so the option A is Linus Pauling. Linus Pauling is model of alpha helix. Then next one is the Fisher. The Fisher, he discovered the lock and key model. Okay. And John Indrew, he has done the myoglobin structure, and Christian and Fincher, which is the option 4, that is the sequence structure. relationship. Which one of the following option correctly matches the content of the column A with the column B? Okay. So the first is A is 2. So the option 1 it is wrong. Also option 4 is wrong. The correct answer it may be the option 2 or the option 3. And option B is 3. B is 3, A2, B3. This one is the correct and this one is wrong. So if you have known the linear spooling model of alpha helix and email pacer, that is the lock and key model, then also you can solve this question. Okay. So the correct answer is option 2, that is linear spooling model of alpha helix and email pacer lock and key model, and John Drew myoglobin structure and Christian Anfinchen, that is the sequence structure relationship. Question number 26. Given below is a schematic representation of southern blot performed to identify single copy integration events of tDNA among 6 transgenic plants. Which one of the following option represents potential single copy events? If there is a single copy integration, then there will be formation of the single band. So let's see where the single bands are formed. A single band, it is found in the T1, and here it is a single band. T2 has the 2 bands. T3 has also 2 bands. T4 has 3 bands. T5 has single band. T6 has also single band. So the single copy events, it takes place in T1, T5 and T6. The correct option is option 1, that is T1, T5 and T6. Question number 27. Autogamy refers to. Okay. So this word is self-defined, right? So what is the answer? Auto means self. Gamy means pollination or the reproduction. So is the option 3. That is the self-pollination of flowers is referred as autogamy. Okay. Then question number 28. Which one of the following condition will switch on lac operon in E. coli? So in E. coli, lac operon is switched on when there is presence of lactose. So option 2 and option 3 both are wrong because in the media there is absence of lactose. Okay. So correct answer is either option 1 or option 4. So now we have to discuss about the glucose. Okay. If in the media there is a presence of the glucose, then the E. coli it will use the glucose bus and the lac operon it will be switched off. So if in the medium the lactose is absent, then lac operon it will be operated. The correct answer is option 4 and option 1 is wrong option. So when the lac operon is on, switch on when in the media there is presence of lactose and absence of the glucose. So the correct answer is option 4. Okay. Which one of the following plant pathogen has largest genome size? Yes, it is a little bit difficult to answer. For that you have to know the genome size. So the genome size of Phytophthora infestans, it is 240 MB. Okay, it is the largest genome of plant pathogen. And Ustilago, it has around 20 MB is genome size. And Botrytis, it has about 40. And Fusarium graminearium, the genome size is 37. So, which one is the correct answer? Which of the following plant pathogen has the largest genome size? That is Phytophthora infestans. Okay, the correct answer is option 1. Okay. The last question, that is question number 30. Okay. A student added DMEM culture medium. Okay, DMEM, it is the Gipko Dalvecos modified eagle medium. What is this? It is Gipko. So student added Gipko Dalvecos eagle medium, culture medium, which was pink in color to growing liver cell. Okay, it is pink in color to the growing liver cell. 3 days later, the medium color was yellow. This indicates, means the indicator, it changes its color from pink to the yellow. So what it indicates? It may be because of the change in the pH of the medium. So let's say the option change in cell morphology. If the morphology of the cell changed, then there will be not change in the color. So this one is wrong. It's because of the change in pH of the medium. Depletion of nutrient in the medium, that also not related to the change in color. Lack of antibiotics in the culture, this is also not related. So the correct answer is option 2, that is change in pH of the medium. So you can solve this question by applying your general knowledge. Okay. In the examination, because of a lot of pressure, we are doing some silly mistakes, and later on we realize that this is very easy, we can do it. It's okay if this time your exam did not go well. So start preparing from now for the upcoming examination. Thank you so much for watching this video, and I'm sure that this video will definitely help you a lot. In upcoming session, we'll solve the questions from 31 to 50 part B of evening session of CSIR NET 2020 examination. If you are new to my channel and have not subscribed yet, then please do subscribe. This channel is absolutely free and I recommend you to go through all the lectures. You will find it's very helpful. At the end, I have solved previous year CSIR NET questions. Bye bye. See you in my next video.